This week, we will start with positive strand RNA viruses. The genome of these viruses are made by sense RNA plus, meaning with orientation 5 to 3 prime. So this RNA can serve not only as a genomic RNA, but also as mRNA. Therefore, whenever this infectious RNA is released into the cell, target cell, it can dock on ribosomes and start translation. All these viruses recall encode RNA-dependent RNA polymerase enzyme, which is very important or required for replication of these viruses. And once this polymerase is synthesized, it starts by the synthesis of RNA minus or anti-genome. RNA minus serve as a template for synthesis of genomic RNA plus. And when we talk about these positive strand RNA viruses, we have to realize that a large number of plant viruses are RNA plus. We will, however, focus mostly on viruses which cause disorders, diseases in humans and animals. And we will start this week with picornaviruses and flaviviruses. And next week, we will discuss togaviruses and coronaviruses. Picornaviruses are non-enveloped. Flavi, toga, and coronaviruses are enveloped. So let's start with picornaviruses. As the name tells you, they are very small viruses. They are non-enveloped. They are formed by RNA genome and capsid. There are hundreds of different viral species belonging to this family, and they have also huge variety of the target organisms. They can infect from humans through birds, fishes, and so on. And also they can cause many different diseases from poliomyelitis through hepatitis, common cold meningitis, and so on. Picornavirus family is divided into 12 genera. However, we will focus on 3 of them, enterovirus, hepatovirus, and rhinovirus. Rhinovirus, common cold virus, hepatitis A virus belong to hepatoviruses, polio, coccidiovirus, enteroviruses. Now, when we look into enterovirus, the two most prominent viruses belonging here are poliovirus and coccidiovirus. They replicate in gastroenteric tract, therefore they must be highly resistant to low pH in the stomach, resistant to enzymes which are secreted in small intestine. And also, since they are enterovirus, they are usually transmitted through orophycal routes. They must also resist environment for a while. And they can trigger differences from poliomyelitis, meningitis, cardiac, and muscular disease. Rhinovirus is a human rhinovirus. It's not just one virus. There are over 100 different serotypes of rhinoviruses, and they cause all common cold. Now, because of this huge variety of different serotypes, it is almost impossible to make vaccine against the common cold. Cardioviruses are mostly infected mice and mammals. They can cause cardiac and neurological diseases. Aftovirus is food and mouth disease virus. It can trigger or cause epidemics in dominant ruminants, and it's highly infectious. So whenever this epidemic starts, there must be quarantine and very important precautions taken to avoid transmission. Hepatovirus, hepatitis A virus, it causes so-called infectious hepatitis in humans, and it's transmitted via contaminated food and water. Be careful. There are hepatitis A, hepatitis B, and hepatitis C virus. All three belong to different families. So picornaviruses hepatitis A, infectious hepatitis. Now, what is the structure of the virus? Virion has icosahedral capsid with pseudo T3 structure. And three protein, VP1, VP2, and VP3 forms this pentamer, where actually it follows this fivefold axis, which are surrounded by this canyon. And this canyon play a very important role in the attachments to target cell receptors. And I stress again, they are non-enveloped viruses. The genome of Picornaviruses is formed by linear single-stranded RNA with positive strand. On the 5' you can notice this VPG protein, which is covalently bound to the RNA. Also, picornaviruses have this very large 5' non-coding regions, which plays very important role in the docking of the viral RNA onto ribosomes. The genome has three regions, P1, P2, and P3, and each one is afterwards when polyprotein is synthesized based on this RNA, each of these regions is cleaved to individual protein. What you can see on 3' and of the genome, there is poly A tail, which is encoded in the virus. So it's not like in human where poly A tail is not encoded, which is added after cleavage. Here, viruses encode this poly A tail. So all genes, as I mentioned before, are translated as a single polyprotein. And this single polyprotein is afterwards cleaved to this region P1, P2, P3, which are further cleaved to individual protein and yield different kind of structural and regulatory proteins. The representative virus of picornaviruses is poliovirus. It is a model virus which have been studied extensively during the development of vaccine. Nowadays, you can hardly see poliomyelitis. That's what poliovirus infection causes, because we have very efficient vaccine. The poliovirus is circulated on Earth for thousands and thousands of years. As you can see on this picture from ancient Egypt, it depicts a person who most probably was paralyzed because of the infection with polio. Also, FDR, our president from 1933 to 45, was on wheelchair because he was paralyzed after polio infection. And FDR, together with his government, supported much of DIME's organization that contributed significantly financially to the development of vaccine. And we will talk a little bit more about vaccine later on. And once poliovirus becomes a model organisms, it actually helps us to develop knowledge on the RNA-dependent RNA polymerases, translation of the RNA plus, and also iris, which we will talk later on about. So how virus replicate? So first, virus has to bind to receptor and enter, uncode and release plastrine RNA, which can directly dock via iris on the ribosome and synthesize the structural protein. When RNA-dependent RNA polymerase is synthesized, it triggers transcription of that plus RNA to minus RNA, and this serves as template for synthesis of genomic plus RNA. Virus afterwards assemble this package and release from cells. So we will focus on each step individually. So when we look for the receptor, we know virus first must interact with receptor. Each virus has a different kind of receptors that used to enter cells, and there are quite a variety of receptor. Poliovirus binds to so-called PVR or CD155 receptor, which belongs to immunoglobulin superfamily. Coxsackie viruses use a so-called death factor, CD55, decay accelerating factor. This is proteins which is expressed on surface of our cells and protect our cells from damage by activated complement cascade. However, they can also use CAR receptor, adhesion molecule, integrins, all kind of different receptor. Rhinoviruses, they use immunoglobulin-like receptor, ICAM adhesion receptor, but they can also use LDL, low-density lipoprotein receptor. Food and mouth disease use heparan sulfate, or they can use also integrins. So there are variety of receptors that can be used by the picornaviruses to enter target cells. Once they bind to the target cells, there are different ways how they enter. Rhinoviruses and foot and mouth disease, the receptor concentrates the virus and trigger receptor-mediated endocytosis. So they enter by a receptor-mediated endocytosis, and they encode in endosome when the pH is lower. Poliovirus and Coxsackie virus actually the receptor function and unzippers, which triggers changes in the virus and disassembly of the virions. Now, this is a model how poliovirus enter. Remember, rhinoviruses enter by receptor-mediated endocytosis. Poliovirus enters by creating the channel inside of cell membrane. So you recall there are these canyons around those fivefold axes, those pentamers, and the receptor interact with this canyon. And this interaction trigger conformational change, which releases VP4, which is internal protein, and VP1 change conformation and extends beyond of the viral capsid, and its N-terminus portion has hydrophobic regions, and this hydrophobic region triggers pore formation. Yes, it forms a pore in the target cell, and this pore enables viral RNA to enter target cell. So this is model poliovirus, which is very well studied. Now look on that canyon and look on this little area here, this little hydrophobic pocket. Basically, when a receptor binds to this VP1, it changed conformation. And this change in conformation is possible because this pocket allowed the change. And it enables tight binding of the receptor at that canyon and triggering those conformational changes. Few years ago, the chemical called pleconaryl has been developed, which fit right in this hydrophobic pocket. What does it do? It stabilizes that VP1, so it cannot change conformation. So it does not allow this tight binding of the receptor with this canyon, and therefore it cannot release VP4, it cannot VP1 change conformation and trigger those pores in target cells. So it blocks entry of many picornaviruses.